Okay, we just went through a whole bunch of great Mendelian inheritance material. So you have seen everything from simple kinds of situations following one locus, so therefore one gene, and we've done simple crosses with that. We've seen some things that kind of go against the normal grain. You know, if you we've discussed incomplete dominance. We've discussed codominance. You should be thinking blood type there. We've talked about multiple alleles. You should also be thinking about blood type there in humans. And then we broke into following more than one locus at a given time. And that is particularly interesting because if they're on separate chromosomes, in which case we call them independent loci, then we have all sorts of fun in sorting out the gametes that each parent could make in following two different loci on different chromosomes. Remember that we created at the end this fantastic kind of 4x4 Punnett square. And then we went one step further and looked at gene interactions between those two loci in a group of phenomena that we collectively call epistasis. And then we had modifications of ratios. Of course, on the note of ratios, you've got some ratios in mind. The most famous one that we've discussed so far is the 9:3 to 3:1, which of course is that dihybrid cross of independent loci, normal dominance or complete dominance, and therefore a dominant recessive allele at each locus. So anyway, that stuff should be going through your mind. You should be pretty solid on it. And what we're going to do right now is actually just work a variety of problems from the problem set that's posted on Blackboard. This is worth money on your punch card. So you have to do the whole set for the punch card experience. There's also a key there. In this particular case, though, we're going to pick out several of these questions and work them right here on the board. The other thing that I want to make mention of right now is that there'll be a lot of these kinds of questions on the final exam. And because of that, and because there's a time limit on it, you're going to want to be pretty good at solving these basic inheritance problems. So let's get at it. So here's a copy of my set of problems, and of course you'll see these in a minute, but there's a total of 24 of these. There's a couple that I'm going to throw out because they deal with linked loci. That would be loci that are on the same chromosome. We're going to stay away from that. You'll have plenty of time to get into that in a future class, but right now we're just going to be focusing, when we consider more than one locus, on independent loci. We're going to start at the top, the easiest one. So this is actually just following one gene. And the number one of the problem set is stated as 2 black guinea pigs were mated over several years. So that's the first thing. That's the first little part of that sentence we want to kind of process in our brain. Two black guinea pigs were mated over several years, produced 29 black and 9 white. The fact that white show up, that should be ringing a bell in our brains. So 29 black and 9 white offspring, the most likely genotypes are. Now, I'm going to put a lot more information to solve this problem. A lot of you could just read this and pick the answer straight up. But I want to write down what I make of the inheritance as I read the question. So we know we have black as a phenotype. We know we have white. We know that these are guinea pigs. And if we mated 2 black, and then we actually see in the ratio that we had in the offspring, we had 29 black and I believe 9 white. That's kind of looking to me like the classic 3-to-1 ratio that we would have expected from complete dominance and a cross of monohybrids. So this is pretty much that. So what I'm going to do is assign alleles. I'm going to give a big B to that and a little b to that. And that's going to be my model. And then if you wanted to look at all the possible answers, that's one of the ways to do this. So we could go with A, answer A is little b, little b, little b, little b. Answer B is big B crossed with big B, big B. You can start to throw some of these out pretty quickly. Answer C is big B, and little b. We'll just work all of these just to make sure. Answer D down here is big B crossed big B, little b. And then answer E is big B crossed with little b. So if these were our parents and these were the correct assignments of alleles, meaning that big B is an allele for black, completely dominant over the recessive allele little b for white, this would be what it would look like. So if this was a cross, let's take a look at what that cross is. Possible gametes for this. The gametes are going to be all little b's from this parent, all little b's from this parent. Now, I'm a reductionist in my Punnett squares. I only like to have the reduced minimum amount of work in a Punnett square. I only need a 1 by 1. I need a 1 by 1 here. So there it is, little b. So I put that gamete there. All of these offspring are going to be that. And of course, they would be all white phenotypically. If we did this one, it's kind of the reverse of that. The possible gametes, this one is only going to make big B, this one's only going to make big B. Again, in the Punnett, I need a 1 by 1. So there's big B, there's big B from that parent. And then here we have the genotype, and all of these would be black. Now, we already know that this is the answer we're looking for, so you can pretty much cross these out right now. Let's do this one. This one's more interesting. Possible gametes here. This is going to produce 50% of a big B and 50% of a little b, and then this parent here, all little b. So I'm a reductionist in the Punnett square, and that means I only need a 2 by 1. So here's the little b, and the big B here, the little b here from this parent. So we're going to get 50% of our offspring being heterozygous, and then on the bottom here, we're actually going to get 50% of our offspring being homozygous recessive. So this would be this is going to be a 1-to-1 kind of, well, the genotypic ratio is big B to little b. That's the GTR. The PTR is also a 1-to-1, and that would be 1-to-1 black to white. Wow. And of course, that's not 1-to-1, so we can cross that out too. Check it out. We're getting through it. Now, this is a lot of work to do for one problem, but I think it's good to do it this way at the beginning if you haven't done these problems before, because you start to really build your technique and your understanding. Because a lot of these questions would be very easy to answer if you have this basis. If you don't, though, it's good to work it. Okay, so here's this one. This is the biggest one. So here we're going to have 50% of our gametes being big B, 50% from this parent being little b. Same for this one, 50%, 50%. Classic 2 by 2 needed. So there's our big B and little b from this parent. There's our big B, there's our little b from this parent. And check it out. You've seen this guy before. This is our classic monohybrid cross. That's the monohybrid cross. Here's what we have. We can get a GTR of this of a 1-to-2-to-1, and that of course would be big B to big B, or little b, and then little b. And then the PTR of this is going to be 3-to-1. That's the big guy. Boom, 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 boom. Classic monohybrid cross ratio with complete dominance. So therefore 3-to-1, and that would be, of course, black to white. Now, of course, even though I'm going to answer the last question, you can see that, wow, this is about 3-1. So this is our answer. This is going to be our answer. That is our correct answer for this question. So number 1, the answer is D. And let's just do this one for the fun of it. Let's get this thing straight. So here, all of the gametes on this one are going to be B. All the gametes on this parent, little b. So big B, little b. 1 by 1 Punnett square. All offspring. All are big B, little b. And the genotype, all are black. Cross that out as a possible answer. So that is way overkill work for number 1, but very helpful to get you into the game of performing crosses. So in questions like these, if you do a cross for every single possible answer, if they are setting up crosses in the answer, then you can really familiarize yourself with this process and get familiar with it. One last note is that you may see in books a non-reductionist Punnett square, in which case anytime you're dealing with one locus you do a 2 by 2. This 1 by 1 business of mine and this 2 by 1 business of mine, it's reductionist because it saves a bit of work. But I think that you might want to see the way that a cross would be presented in the text. For instance, if let's look at this one right here. We're going to look at this one right here as it might be presented in the text. Again, I'm out of the question now. We're no longer answering question number 1. I'm just going to show you the way that the book would do that Punnett. The way that the book would do that Punnett square would be, oh, well, that's not a very good one, but they can do it. It would be like that. What your book would do would say the possible gametes of this parental cross would be little b, and then little b, so all little b's, and they would actually draw this as a 2 by 2. And it's the same answer, isn't it? All white. All white, and I guess genotypically they're all little b, little b. So I'm just going to let you know that that's the way you'll see it. So anytime that you would do single locus inheritance in the textbook, you're going to make a 2 by 2 no matter what. For me, you'll make a 2 by 2 sometimes if needed, but sometimes you'll make smaller than that because, again, we're only dealing with creating rows or columns in Punnett squares for unique types of gametes. So if you've got one parent that only produces one type of gamete, that parent only produces one row or one column in a Punnett square for the reductionist method. And that's how I do it. Okay, moving on. Let's keep it straight and simple at the top. We're going to do number 2 because number 2 has different kinds of answers in the problem solve. It's not possible. Parents and genotypes in business. This question number 2 is in humans. We're going to actually oversimplify human eye color, by the way. We're going to make it real simple, and that's what this question does. So in humans, brown eye color is dominant, blue is recessive. Wow, I need to write my information down right now. So in human, brown eye color is dominant, so I'm going to give it a big B. Blue eye color is recessive. So do you see how I wrote that first sentence right there in our code? If a brown-eyed man, here's our cross then. So I'm going to put a male for man, and I'm going to say brown, and I'm going to put blank for question mark. That is a brown-eyed man of unknown genotype. So a brown-eyed man marries a blue-eyed woman. Now that's easy. So I put female up to indicate woman, and then I know that if she's blue-eyed, according to everything given to me, that she's clearly homozygous recessive, right? And that's the parents, and in the F1s, here's what they have. In the F1s of this cross, they have a brown-eyed boy, and we know that the only way to get a brown-eyed boy would be this way and this way. That's the only kind of gamete you can get there, and at least the brown-eyed parent, so they have a brown-eyed boy. That's not very informative. It doesn't help us answer this question. We are really all about, we want to know what the heck this guy is right here. That's the whole thing. So then the next part is, the other offspring that they have is a blue-eyed girl. Oh, that does it. So here, if they've got a blue-eyed girl, that's her genotype. And it's this one right here. It's that one right there that had to be a recessive allele. If you make blue-eyed offspring in this cross, that parent that's brown-eyed has to be heterozygous. That is the only way that it can happen. That is very cool. Okay, so then we can look at our answers. Answer A, the children are adopted. Yeah, sometimes we have silly answers like this. Knock it out. That's answer A, out. So answer A, you can read, boom. Answer B, the man is heterozygous. Well, that's clearly the answer. That is the one that we go with. So basically, the man is heterozygous. That's what we just determined. You can read the other ones. Eye color is sex-linked. I haven't even talked about sex-linked. I would avoid that kind of question on the final since I haven't talked about sex-linked. Basically, for those of you that have these inquiring minds, sex-linked traits or sex-linked loci occur on the X chromosome. So I'll say that one more time. Sex-linked loci occur on the X chromosome. Now, when you get into solving problems with sex linkage, then you'll know that females could have two versions because they've got two X's, and males only one. That's a curveball. Again, there's great times in the future for you to get into sex-linked chromosomes. A lot of cool things happen there, but for us, we don't care about that right now. Next one is C. That was C, so that sex-linked thing was out. Answer D is that both parents are homozygous, and of course that's one that you could actually do the cross. There's no way to get a brown-eyed boy here, right? No way to get a brown-eyed boy there. So that one gets thrown out, and then finally, here's the crazy one. The man has the genotype for blue eyes, but the phenotype for brown eyes. And that's not working in the way that we've presented eye color. As a matter of fact, that answer becomes much more tangible in terms of real human eye color because of something called modifier genes, but that's not the scope of this problem. We're keeping it simple. One locus for eye color, two possible alleles, big B for brown, little b for blue. All right, so that's number 2. Very cool. You can see two different kinds of questions then. There's questions that actually show you genotype answers. There's questions that show you kind of written answers. And then another question that we could do will get us into another kind of answer. And so that will be number 3. And in number 3, we're going to one of our main systems in the problem set. In number 3, we're going with garden peas. So what we're going to say here is in garden peas, tallness is dominant and dwarfness is recessive. Tallness is dominant and dwarfness is recessive. We've already used the word recessive and we've used the word dominant. So we're going to say tallness is going to be big T, and then dwarf, they call it dwarfness. What a great word. That is a really great word. Dwarfness. Anyway, tallness and dwarfness. So those are our alleles that I've already assigned using our standard code for alleles. Of course, remember that later on when we get into things like incomplete dominance and codominance that we use different systems, like primes and things like that. But for normal dominant and recessiveness, it's cap in lowercase. All right, so now we got that. That's our first sentence. A heterozygous tall plant, wow, big T, little t. So that's the first part of the second sentence. A heterozygous tall plant is crossed with a dwarf plant. So I've laid out that cross right there. And here's what we want to do. If 40 offspring are produced, the number of tall individuals will be. So this is our third kind of answer profile to show expected offspring from a cross. So we know that the gametes here would be big T and little t. We know that the gametes here would be all little t. So we're going to need a 1x2 or a 2x1, I guess we could call it. And there's big T, there's little t gamete, and there is little t. So we're going to end up here, and we're going to end up here as our possible offspring. So our GTR is going to be 1 to 1, and that's going to be big T, big little t, big T, little t to little t. And then our PTR, phenotypic ratio, is going to be also the same, 1 to 1, and that would be tall to dwarf. So there it is. So we already know then how to solve this. So of your possible answers, we're looking for out of 40, how many would you expect, by the way? We always have to honor that this isn't absolute. We're playing basically probability here, just like we did back in meiosis with coin tossing. But anyway, how many would you expect to be, I think it asks for tall. And so we know that half of them would be tall. So without even doing any of the math, 20 of them would be tall. And there's that answer. So that answer sits at C. Answer C. So there you have 3 different profiles for the types of questions and the types of answers that you get. Let's see now. What should we do? Ooh, that's exciting. How about doing number 5? Number 5 is going to fold in some of our other concept. Okay. Number 5. Now there's a lot to say here. You're going to be able to see these questions here when I show the screen and read them. So here we have it. Mendel used pollen from a true breeding dwarf strain of peas. Okay, so I'm gonna say that we've got dwarf. True breeding equals homozygous, so it's really good for us to use Mendel's language and be familiar with it and how it relates to the modern language. So dwarf strain, true breeding. We're probably going to keep consistent with what we've read about before. These were peas. We already did a pea question, so we already know that dwarf was little t. And we're not going to change that. So in other words, if you see a P question on the final exam, tallness is dominant, dwarfness is recessive. Okay, but anyway, we're gonna go with that right now. And of course, it's obviously true breeding. In this case, means homozygous. But the only way to be dwarf is to be homozygous or true breeding. So we have dwarf plants that look like this: little t, and he used those to deal with a true breeding line of, so this is true breeding, and we'll use those to deal with a true breeding line of tall, true breeding tall. And so what he did in his cross was he took this and he did it with this. So there's his parents. Obviously, if you want, here's your gametes. All big T, all little t, all big T. For me, it's a 1x1. And that's how this looks. All of the offspring, all of them are tall. And then we're going to go on here he planted the resulting seeds. So these are seeds. He's just going to kind of, they're just going to indicate what I've already put on the board. He planted the resulting seeds to find that only tall plants grew from them. Check. Because the only way that you see tall is to actually plant the seeds and grow them up. And then he used the pollen from these tall hybrids to fertilize the flowers from the original dwarf. So he took one of these then, and he used those in the original dwarf. So here's the original dwarf right here. Now we have to set up this cross and we can get our answer. So we set up this cross. Possible gametes for this experimental cross, 50-50 big T, little t. All of these are little t. That, of course, is also our familiar reductionist Punnett square of a 2x1, big T, little t, little t, little t. Genotypic ratio is 1 to 1. 1 to 1, big T, little t to little t, little t. Phenotypic ratio is a 1 to 1 in this cross expected. Tall to dwarf. Not dwarf. Dwarf. Okay, so there we have it. Now we already know that 50% of them are going to be it. Now we can answer the question. So first off, these are different answers. The first answer, A, is that it's monohybrid inheritance with codominance. That's probably not the best answer. Codominance would mean that, remember that codominance, like blood type, the classic demonstration of codominance is in type AB blood, where the heterozygote expresses both phenotypes from each individual. We also talked about codominance in the hemoglobin phenomenon in the sickle cell case. Remember the HB allele? So we have this going on. Very interesting. That one doesn't go. Codominance is out. Monohybrid inheritance with partial dominance. Well, partial dominance would produce an intermediate phenotype, which would be like mid-sized in the heterozygote. So here, what this question is doing is it's making you just kind of throw out some other concepts that actually have a whole different kind of inheritance predictions. And then or C is monohybrid inheritance with simple dominance. Yes, that's our answer. I would say complete dominance, but with simple dominance. And then we know it's not dihybrid. That's answer D and E because we're not following more than one locus. So here's a curveball for you. When I say monohybrid cross, I mean that. I mean crossing two monohybrids. Monohybrid inheritance. Monohybrid inheritance, as used in these Mendelian questions, involves at least one monohybrid. That's kind of how it works. Because never did we actually see a monohybrid cross here, which produces the 1 to 2 to 1 genotypic ratio and the 3 to 1 phenotypic ratio when you have normal or simple dominance as stated here, so what I call complete dominance. So there you have another kind of question where the answers are actually making you cast aside other inheritance systems, other inheritance concepts to get at the heart of the matter. For this one, you needed to be able to perform the cross, but you really needed to be able to understand the difference between monohybrid and dihybrid, and you needed to know the difference between simple dominance, incomplete or partial dominance, those are kind of synonymous, and codominance. You needed to know all of that stuff to get the right answer. So these are 4 classic examples of simple inheritance. We've only got one more to do, focusing on one locus, and that would be when we have multiple alleles. And so we're going for a blood typing question with humans, and it's a lot of fun. Okay. Here we go. We're going all the way to the very end, number 24. This is a human blood typing question, so you should immediately be thinking, oh, okay, we got cap IA, cap IB, and little I. We got multiple alleles. We have codominance between cap IA and cap IB, all that good stuff that we kind of talked about before. Now you get to solve a problem. So number 24 is a woman. So I'm going to put female. And I'm going to say with type AB blood, I super A, I super B. Marries and basically, I guess we're going to assume they're going to reproduce, but at this point they're just married. It's going to be a male, and this is a male that's type B blood. Remember that this is a blank. It's not really a complete blank because it could be IB or little I, right? There's two genotypes to be type B. So that's what we've got. And then one of the man's parents had blood type O. Oh my God, that's all I need. So this guy had, one parent was little I. Therefore, we know that this is the case. Let's see. So this is little I. So one of these guys' parents had this genotype, and that's how we figure out that this is little I, because that parent could have only given this guy, the husband, a little I. Now we have the whole thing set up, but they want percentage for the question, so we have to do the cross. So here's our parents. We set that up correctly. We have big I super A, we have big I super B, so 50% of gametes from the female, from the mother over here, and then we're going to see also a 50-50 here. So we're going to get 50% of our gametes being I super B for the father, who's type B blood, but he's heterozygous, and therefore 50% of the gametes are going to be little I. And then this is us brewing up a beautiful 2x2. So there's I super A, there's I super B, there's I super B and little I. Let's bring them together, come up with predicted genotypes in the punnet. There's that. Look at this, homozygous codominant, anyway, type B. And boom. Now we have a really fun ratio to sort out. Here's the GTR. The GTR actually is, they're all unique. So the GTR is a 1 to 1 to 1 to 1. How cool is that? And of course in that, it's basically an a type AB, whoops, comma, or actually to IA little I, to IB, IB, to IBI. So that's the genotypic ratio, 1 to 1. But the phenotypic ratio is really fun because we have one type AB, we have one type A, and beautifully we end up with 2. So this is a 1 to 1 to 2 ratio of type AB to type A to type B. A 1 to 1 to 2 off ratio. So then, of course, here's our answers. Theoretically, what percentage of the couple's children will be type B? We already know that. So 50% is what this is. 50%. You can see it right here. So that's 25%, 25%, 50%. 50% of the offspring. Your answer sits there as C. There you have it. Now you've seen 5 beautiful examples of kind of one locus inheritance. I guess the only one that I didn't show you involved partial or incomplete dominance, which of course changes phenotypic ratios. I'm not going to do that in a one locus system. I'm going to do that in a two locus, independent loci, that is, system with another problem. So now we're actually going to blow it up one more step and start to consider another gene on another chromosome. Excellent. Okay, now I told you we're going to step it up. We're going to bring it in two different loci, independent of course, on separate chromosomes. We're going to look at number 19. And 19 is a great question. It's talking about cattle. That's going to be our system, and let's read this. In cattle, there's an allele for horns that is recessive to allele for no horns. So therefore, big H equals horns. Is big H equals no horns. Little h is equal to horns. So this is one of these curveballs right off the bat. You're thinking, oh, horns, that's got to be dominant, right? In this case, no. So actually, the only way to have horns is to be a homozygous recessive, as you would predict. So we already know that it's recessive. So that is the first kind of sentence, all right there. Cattle, this means no horns. This means horns. It's just great to take these sentences and put them into the straight kind of symbolic language of inheritance problems. That's what I'm trying to train you. Okay. Then we go for coat color. So we have big R is what we would call a red coat. And then little r is actually for a white coat. And the heterozygote here is equal to roan. Now, first off, I would opt for assigning different alleles, but that's not in the question. You know, these things are coming from databases. They've been made. This is a 20th century genetics inheritance problem question from a textbook that we don't even use. It's not even published anymore. So here we have big R, little r. I think what I would do is maybe go with big R and big R prime. This would be like a kind of preferred Hancock, whoops, I can't even spell my own name in tight spaces. Hancock preferred would be to actually use, because what we're talking about here, you guys, is that roan is intermediate. This is an intermediate phenotype for a heterozygote. So we call this incomplete dominance. Incomplete dominance. Or in the other language that shows up, partial dominance. These are both things that will show up in your lingo. And this is so at this particular thing, we have that example. Now, we can't change the alleles. This is the way they're published in the question. So, but this indicates incomplete dominance. There's no such information given for horns. Like you don't have like medium horns if you're heterozygote. It's basically no horns or horns. So here we have two different things to follow. And continuing to read, what I need to insert, and I would insert if I wrote this question myself, is assume, this is a Hancock insertion, assume that the loci for kind of horns and coat color are independent. That's one way to write it, which would be equal to are on separate chromosomes. Okay? Very important for you to understand that. Anything that I'm going to throw at you when we consider more than one locus in this particular section of our course is going to be, they're going to be independent loci. I'm not going to deal with anything with linked loci, either just regular linkage or sex linkage. So Linkage is not for you for right now. You might see a couple of questions on linkage and you should actually crack into the book and answer them for fun. But for us, we're just going to focus in developing our skills for independent loci when we consider more than one locus. Okay, so we've got that. Let's now get the punchline here for setting up the cross. So, still working on 19, by the way, number 19. And here's the punchline then, is if two roan cattle, here's our cross. So two roan cattle, heterozygous for the H allele. So we already know that they're heterozygotes. Would these have horns or not? I hope you answered that in that short break. They don't. So that, and then roan indicates that. Oh my, look at what we're doing. We're setting up the classic dihybrid cross. Dihybrid cross, fun times. So you guys can do this either way that you want. Here's the old school long way. The long way is, this is a dihybrid cross. You want to know what to do. You're going to have to perform the cross. So here's your parents. Here's your possible gametes. So we have to have a gamete from each of the loci. So we have that. We have that. We have that. And we have that. 4 gametes from that parent. 4 gametes from this parent. And we have these going in the same order because what we want to do, therefore, we need a 4x4. And let's get on that party. It's good for you to see another one of these. We did this before. We'll do it again. So let's run our gametes. So this parent's gametes in our 4x4. I guess I should circle these. That's kind of been my code for gametes. And therefore, we'll circle those. You always want to kind of have a basic fundamental system. These parents are going here, and you want them to go, and when you have a classic dihybrid cross like this, you definitely want to run them in the same order. Down for the left parent and across for the right parent. So we've got this, and we've got this. So just start filling them out. Big H, big H, big R, big R. It's kind of interesting if you do it this way. You might see this trend. I'm going to show you right here that anytime that you work on this diagonal, you're going to get the classic kind of dihybrid all the way across. That's very cool. So I'm just going to fill that in right now because I already know what's happening because I did it right. Now, of course, if you're doing this and you have time on an exam, don't go to any shortcuts like this. Make sure you follow it correctly. Okay. There we have it. Beautiful, beautiful. Of course, what you want to do is process this in terms of ratios. So here's going to be, the first genotype is going to be this one. Big H, big H, big R, big R. And we have one of those. So I'll put a tally mark there. Then I'll write this next one. Big H, big R, little r. We've got one of those. I'll tally that. Here's yet another one. So if it's a novel genotype, you write it down, and if it's not, you just tally it. Here comes that dynamic kind of dihybrid. That's our first one of those. You see what I'm doing? We did this before. Here it is again. So here we go. We've already got this one right here. Do you see that? Here's big H, big R, little r. There it is. Tally and click. This is new. This is the dihybrid that we saw. Boom, boom. This one is new. Big H, little r, little r. And then this guy we've seen before, right here, check mark and tally. This guy we've seen before, check mark and tally. This guy we have not. Little h, big R, get boom. And then this guy right here is little h, heterozygous. Haven't seen that yet. This guy here we have for sure. That's that one. This guy here we have there. This guy here we have he. And then finally the last one is little h, little r, tally, boom. So you should add this up to 16. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. We know that we've done this correctly then. Phenotypic ratio, let's take a look at this. This is where it gets a bit fun. That's what we're trying to do. So remember that this locus here is the incomplete dominance locus. So this is going to be pretty interesting if we kind of take a look at what happens at the R locus. So here is a no horn, and then big R. We erased what this is, but this is no horn. This is no horn red. So no and red. This one here is no. If it has a big H, it's a no. And roan. Ooh. This one is no and red. This one is no and roan. This one is no and white. This one is no and white. This one is horn and red. This one is horned and roan. Oh my. This one is horned and white. Okay, so what we're looking for then, now that we've done all of this work, is it the answer? We have the question. The question is, what kinds and ratios of offspring do you expect? So let's identify these. So we have here. I'm gonna erase the board so we can actually start to deal with our phenotypic ratios. Let's see. I'm going to take the cross away because we've got the cross summarized. Got the cross summarized, and then we can actually find out what these are. So this one here and this one here are going to be no horns in red. And then this one here and this one here, no horns and roan. And let's see. So let's see. Let's do this. I'm going to draw my arrows and then assign them. Here's no horn and white right here. And then let's see, it's going to make sure everything's okay. We're getting there. Use blue for this one. This is horned and red. That's all we got. This one is horned and roan. And this one is horned and white. Wow. Okay, so let's write them down then. So we have a total of 3 no horn reds. We have a total of 1, 2, 3, 4, 5, 6 no roan. We have a no white, 3 no whites. We have 1 horn red. we have 2 horn roan. This is crazy. And finally, at the very bottom, we have 1 horn white. So can you find an answer that has a ratio like this? Some of them you can toss out if you did the whole thing. Any of the ones that have a 9, you're going to toss out. And so B, C, and E, if you look at those, have 9s. So let's confirm that A is our answer. 3 hornless red. So 3 of those, that's check mark. 6 hornless roan, check mark. 3 hornless white. Let's see, no horn white, check mark. 1 horned red, check mark. Okay, 2 horned roan, check mark. And 1 horned white, check mark. Our answer, beautifully, is A for number 19. Man, that's good time. That's big work. It's an exam, and this thing is worth 2 points, just like the previous one. And the first one, doing it took you 30 seconds. This one took you 4.5 minutes. If you do it beautifully like this. Now, if you do the full cross like this, it gives you great confidence. And I want you to work these full crosses to get experience as you go through the problem set. But there's another method for this, possibly. Let's see. Matter of fact, I think the best way to do this is the full cross. Just the full cross. So that's all. I'm just going to leave that one on the table like that. That's good stuff. I'm going to take you into a time-saving method with multiple alleles, with multiple loci. That actually is kind of fun. It'll be the last question that we deal with. Right, we're going to do, now we're going to consider more than 2 loci. They're going to be independent. It's big, it's exciting stuff. I'm going to take you down the path of number 22, and in number 22, we're going to do what they're calling a tetrahybrid cross. It's not really a tetrahybrid cross, but they're going to set it up like this. So notice hetero, hetero, homo, homo for that guy, and then this guy here is going to be the complete quadrahybrid. That's what that guy is. Now, if you're going to set up this cross, it's going to be pretty crazy for you to do as a Punnett square. If you followed kind of the classic determination of how many rows and cells, you weren't reductionist, you'd need a 16 by 16, and that's a lot of real estate. You don't want to go there. So what we really want to do is isolate each of the loci together. So, and then we're going to multiply all those independent probabilities. So we're going to set up actually 4 little crosses, and I'm doing them with colors so you can see what is going on. So this guy then, for the first one here, we're going to set up a little a, a big A, little a. So this is your classic kind of, we're going for, man, this one's too easy too. Crazy. I wish we had a better question there. That's a stinker. All right. You know what? I'm going to answer this darn thing. Okay, so this guy would be, from that one, you want to know what proportion will have the recessive phenotype. So proportion recessive, question mark. This guy, if you did this cross, you would remember that 1/4 of them did. Anyway, if you did this cross, it's this. And then, of course, what we're looking for is that, which is, actually I should stay away from that color, that, which is 1/4 of the offspring, or little a. And then here for this guy, for B, it's going to be exactly the same. So same as that, and therefore 1/4 little b. And then we go to the next guy, and the next guy is right here. This is where it gets crazy because big C by this. Here you already know that this produces a 1/2 Punnett square. It's supposed to be little c and big C. So here you have 0. There are no recessive phenotypes there, so I already know the answer to the question. This one's exactly the same here. Let's see. Oh, come on. Big D, little d. So same kind of thing. Big D with a 1/2 Punnett. This parent, big D, little d. Do the cross, and we'll see also 0. So the answer to this question is none. None of them are what we would say little a, little b, little c, little d. That's what the question's asking for. So technically speaking, it's none is grammatically. Anyway, none is. So that's the answer to that particular question. It's not the greatest one in the world. I'm going to do one more that actually will allow us to kind of set the whole stage. And it's pretty complicated. We're going to back up 1 on the sheet and do number 20. So here comes number 20. All right. So for number 20, lots of info to put at the top. So we're in garden peas. So we have big T equals tall. We have big S equals smooth. We have big R equals red. That's flowers, that is. And A equals axial. This is the classic Mendelian toolkit right here. We got it all. And they are dominant over their respectives. So this was dwarf, you might remember. This was wrinkled. This was white. Let's see, and it says that. White. And then finally we had lowercase a for terminal flowers. Terminal flowers. So these are two dealing with flowers. This is seeds, this is the plant itself. So there we have it. Suppose that two individuals that are heterozygous for all 4 are crossed. So I'm setting that up. Now, as mentioned before, you want to do that cross, that's going to take like at least half of your exam period because it's a 16 by 16. I'm just going to tell you right now, and that is crazy. So we've done 4 by 4s with 2, add a third, it's 8 by 8, add to a fourth, and it's 16 by 16. That is crazy. So you're not going to do it that way. You're going to focus on each individual one to determine what happens. Here's what they want to know. Here, they want to know this question. What proportion will have the dominant phenotype for all 4? So the way that we do this is independent probabilities. So we'll start with this one. So in this particular one, we set up individual crosses. We know, therefore, that if this is one independent event, because that's only on one chromosome. That's on another chromosome. That's on another chromosome. That's on another chromosome. These are independent loci. So these are all separate coins, is basically what we're talking about. And therefore, what we want to do here is set up the possible gametes from this one. You know this. So big T, little t, big T, little t. This gives us the classic 2 by 2 big T, little t, big T, little t. Do your business. And then what we want to do is answer the question. What proportion of this will be dominant? We can see that these ones, these ones here are dominant. 3/4 will be dominant. And then guess what? All of these are the same. So then we do the S's, and we end up 3/4 are dominant. Then we do the flower color, and 3/4 of these are dominant. And then finally we do the, let's get a better color. Let's see. I guess, how about purple? So here we do the flower position, and it's 3/4. So basically what you guys are going to do is multiply these three to get your answer. So that's 16, 64, 128 is our denominator. And then 3, 6, 9, 27, and 67. Let's see. Did I do it right? 4, 8, 9, 64. Oh, whoa. 256. 256, and then we end up with 81. 81 over 256 is your number if you do that math. That is, for this particular question, answer C. It really seems like way too many of these questions in this set have C being the answer, but that's okay. Work it, do it. This is crazy. There will be a couple of these kind of more than 2 loci questions on the final, so you're going to need that shortcut. It's basically independent events. If you solve, you got to find out what the question is, and then basically solve independently for each locus. These turned out to all be the same because these were each quadrohybrids, true quadrohybrids, and therefore it's going to be boom, 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 boom like we saw, and that's quite spectacular. It's good stuff. So there you have a very, very good set of experiences to work from in terms of these problems. We've done a chunk of these. To get the full credit for your punch cards, you're going to do all of them. And of course, I do have an answer key. It's a simple answer key. It's only the letter of the answer. So if you got it wrong, you better go back and figure out why you got it wrong. All right. So that is all I'm going to show today. We got 3 more lectures. In the 3 more lectures, in these 3 lectures, we are going to get right back into DNA. We're going to pull up the structure of DNA. And we're going to identify the numbers of carbons of the sugar. And with that, we're going to identify that a strand of nucleic acid, DNA or RNA, has what we call directionality. We have a 3' and a 5' that are part of the sugar phosphate backbone. We're going to be able to identify nucleic acids in direction as being 3' to 5' or 5' to 3'. We'll describe processes that occur in terms of 3' to 5' or 5' to 3'. Once we have that under our belts, you're going to be able to clearly understand the whole process of DNA duplication, and then later the magical process of transcription to make a messenger RNA, and then the messenger RNA goes out into the cytoplasm and does its thing. Passes through a ribosome, which is also made of RNA, and carries with it this 3-unit code, 3 bases at a time that code for specific amino acids, obviously then making a chain of proteins, primary structure of protein, like we talked about a couple of months ago. Wow, wow, wow. Things are exciting down the home stretch, so I hope that you're enjoying these lectures, and that's all I got, man. I'll see you on Thursday.